Okay, so our topic is the molecular basis of genetics and gene expression. I divided the lecture into two parts. The first part we shall be dealing mainly with molecular basis of genetics. So here we'll have a brief review of the structure of DNA, followed by a discussion of the central dogma, which is really the basis of our lecture for today. Then we'll be discussing how DNA is polymerized. Now, understanding the principle behind the polymerization would allow you to fully understand replication in both prokaryote and eukaryote. So now let's have a brief review of the structure of DNA. So DNA structure is shown here. DNA is a double-stranded polymer of nucleotides. So this is the first strand with an anti-complementary strand bonded to it. Now, if you take a look at the individual strands of a nucleotide, a DNA strand is composed of nucleotides that are linked together by phosphodiester bond. So the individual nucleotides in turn are composed of the base that is bonded to the sugar, which is deoxyribose, which in turn is bonded to a phosphate. So let's take a look at the unit of a DNA polymer, and we refer to that as a nucleotide. So the nucleotide is composed of the base, either a purine or a pyrimidine, and this is glycosidically linked to the sugar, which is deoxyribose. Deoxyribose is a 5 carbon sugar, and the carbons are numbered as such 1 prime, 2 prime, 3, 4, 5 prime, and so on. And we always put the superscript prime to differentiate them from the carbons of the base. To carbon number 5, linked to that is a phosphate group. Now, nucleotides are linked together by phosphodiester bond. Thus, a DNA would have a sugar and a phosphate as its backbone. Now, as you can see here, DNA has a polarity, which means it has a head referred to as the 5 prime end. Now, this 5 prime end always bears a phosphate. And the reason why we refer to it as the 5 prime end, because what is sticking out here is the 5 prime carbon of the sugar. The tail is the 3 prime end, which always bears a hydroxyl group, very important for the formation of phosphodiester bond. Now, the reason why we refer to it as the 3 prime end is because what is sticking out here is the carbon, 3 prime carbon, that bears the hydroxyl group. DNA is always double-stranded, so there is a complementary strand that is running anti-parallel to it. And those two strands are held together by hydrogen bonds between the bases, 3 hydrogen bonds between G and C, while there are only 2 hydrogen bonds between A and T. Now, the number of hydrogen bonds and the composition of DNA would have a profound effect on its melting point. So if there are more G and C's on the DNA, there would be more hydrogen bonds and it will take more energy to separate the two strands from each other. Thus, they have a higher melting point. In contrast, if A and T are predominant, like in this example, there would be relatively less hydrogen bonds. It would be easier to separate the two strands from each other. Thus, they would have a relatively lower melting point. Now, since DNA has a polarity, the sequence of the bases on the DNA should always be read in the 5 prime to 3 prime direction. Thus, this strand sequence is read as G, T, T, while the complementary strand sequence is also read in that direction, A, and C. Now, what is the function of DNA? We all know now that DNA is the genetic material, and the central dogma, which illustrates the function of DNA, actually describes the flow of genetic information. This central dogma was originally proposed by the tandem of Watson and Crick. For those who do not know, Watson and Crick is the tandem of scientists who elucidated the structure of DNA. aside from elucidating the structure of DNA, they also propose that DNA is the genetic material. Sounds like a no-brainer for us now, but back in the 1950s, when they proposed the central dogma, a lot of scientists believed that protein was actually the genetic material, which turned out to be incorrect. Very few scientists believe that DNA is the genetic material, and one of them is Watson and Crick. Turns out that they are correct. Now, according to them, the genetic information within the DNA can be accessed by using DNA as a template to synthesize RNA so that the information can be transferred into the cytoplasm, while RNA is used as a template to synthesize protein, a process known as translation. So, in other words, the sequence of amino acids in the protein are actually determined by the sequence of bases that are located on the DNA. What's great about DNA is that DNA can also be used as a template to synthesize an identical DNA. That process is what we call now as replication. And this DNA, which is identical to this, can be passed on to progeny cell, bringing with it the genetic information that was originally found on the template. If you notice, the original central dogma is missing one process, and that is reverse transcription, which is a late addition. It was only added in the 1970s with the characterization of retroviruses. As you know, retroviruses are viruses that use RNA as genetic material, and they are capable of synthesizing DNA using RNA as a template. We refer to that process now as reverse transcription. As you will see later on, retroviruses are not the only ones that are capable of reverse transcription. Even our cells, to a limited degree, do perform some sort of reverse transcription. Now, let's discuss how DNA is polymerized from nucleotide. Understanding the principle behind this would allow you to fully understand DNA replication itself. Polymerization of DNA follows a certain principle. It will only proceed in one single direction. We refer to that as a 5' to 3' direction, which means we can always add a new nucleotide at the 3' end, but never at the 5' end. So in simple terms, it is the tail that grows during polymerization and not the head. And what is the reason for this? 3' hydroxyl group is an absolute requirement for the formation of phosphodiester bond. As a matter of fact, the enzyme DNA polymerase cannot synthesize a phosphodiester bond de novo. That means from nothing. There should be a pre-existing 3' hydroxyl group. Thus, primers, which are composed of RNA, has to be already present before DNA can be polymerized. It is an absolute requirement. Now, to determine the sequence of the new strand being synthesized, there should be a complementary DNA that would act as a template, and the sequence is determined through the usual base pairing. The enzyme that catalyzes the process is DNA polymerase, and of course, magnesium acts as a cofactor. To illustrate this principle, we have here a simple diagram. This, of course, would act as the template. Take note of the polarity. This would be the sugar phosphate backbone, while this is the 5' end, and this is the 3' end. Our goal is to synthesize the new strand represented by this. As you can see, we do not start from a blank slate. There should already be a pre-existing oligonucleotide in the form of a primer. Now, to incorporate the new nucleotide, we simply follow the base pairing rule that is dictated by the sequence of bases on the template. So on the template, if we have a C, a nucleotide triphosphate that pairs a G would come in. Well, the substrate should be a triphosphate because it contains enough energy to drive the reaction forward. Now, this will be catalyzed by the enzyme DNA polymerase with magnesium, which is positively charged, acting as a cofactor. It will be attracted to the oxygen of the phosphate, which, as you know, is electron rich. Now, because of the attraction of magnesium to the oxygen, there will be uneven distribution now of the electron cloud, such that the phosphorus of the phosphate will be depleted of the electron cloud. In turn, it will have a positive charge. Now, this 3' hydroxyl group's oxygen has an outer electron pair on its surface, just like all other oxygen. So this is negatively charged, positively charged. The attraction is so strong that this electron pair will launch what is known as a nucleophilic attack. Sa Tagalog, ito yung pag-ibig na may halong libog. The attraction is so strong that you want to attack. This results in the formation of a covalent bond that incorporates that phosphate to the formation of the phosphodiester bond. At the same time, there will be hydrolysis of the inorganic phosphate, causing release of energy that drives the reaction forward. Now there's a new 3' hydroxyl group that will launch the nucleophilic attack. So if you notice, the growth of the DNA is from this direction, growing towards this direction. This is what we call a 5' to 3' polymerization, where the 3' hydroxyl group is the only one that can accept an incoming nucleotide. So remember this principle, and you would understand right away why there is a leading strand and a lagging strand in DNA replication, and why there is shortening of telomere, and as well as in the lengthening of the telomere. Let's go to DNA replication itself, starting from prokaryote. Now, prokaryotic DNA is double-stranded. So the 2 parental strands will serve as template. Each of them will serve as template. Thus, they have to be separated from each other by the enzyme helicase. So the 2 parental strands will be separated from each other, as each one would act as a template. The separation is accomplished by the enzyme called helicase that breaks the hydrogen bond between the 2 parental strands. Now that we have separated, each parental strand can serve as a template to synthesize the new strand. The process of replication is catalyzed by 2 enzymes, DNA polymerase III and DNA polymerase I, with DNA polymerase III being the main replicative enzyme. And the process is said to be semi-conservative. What does that mean? We'll take a look at the parental strands. They have been separated from each other. That means they have not been fully conserved, but they have not been destroyed either, but rather they are base paired with a new strand. That means the process is not dispersive nor conservative. It's somewhere in between. We refer to that process as semi-conservative. We can divide replication into 3 phases to allow us to follow the events. The first is initiation, followed by elongation and termination. Initiation refers to the process where the parental strands are separated from each other. We can also refer to it as unwinding. And of course, like what I said, this is catalyzed by the enzyme helicase. This would be followed by laying down of RNA primers. After the primers have been laid down, we shift to the next phase, which is elongation. So here, oligonucleotides can be incorporated into the 3' hydroxyl group of the primers, and actual DNA can now be polymerized by the enzyme DNA polymerase. Proofreading is very important for this phase as it ensures fidelity of replication by removal of mismatch bases. And when the entire DNA has been replicated, that's the end of the process. Let's start first with a simple version of replication. So let's say this is a closed circular DNA found in prokaryote, and of course, it is double-stranded. The process of replication starts here. We call it the origin of replication, or ORIC for short. So there will be separation of the 2 parental strands as catalyzed by the enzyme called helicase, represented by the yellow triangles. Now that we have single strands, each parental single strand can serve as template for the synthesis of DNA. But first, primers have to be laid down, represented by the green bars. What are these primers anyway? Well, primers are not composed of DNA, but rather composed of RNA. These are RNA oligonucleotides laid down by the enzyme primase, and their function is to provide the 3' hydroxyl group that is very important to initiate DNA synthesis because, like what I said earlier, the enzyme DNA polymerase cannot synthesize a phosphodiester bond de novo. After the primers have been laid down, the 3' hydroxyl group at the end of the primers will be targeted by DNA polymerase to synthesize now DNA in the 5' to 3' direction, 5' to 3' direction. So there are two main enzymes that carries out replication, DNA polymerase 3 being the main replicative enzyme, and DNA polymerase 1, whose function we shall be discussing later on. Let's go now to the full details of the process. Let's imagine this as a closed circular double-stranded prokaryotic DNA. The process of replication starts here. We call it the origin of replication. So let's say we have a magic microscope that can zoom in into the origin of replication to allow us to view the events that take place there. So this is the origin of replication, a segment of the closed circular DNA. Take note of the polarity of the two parental strands. So the first event is that the two parental strands will be separated from each other by the enzyme called helicase, as represented by the orange triangle. Now, this single-stranded DNA has still the tendency to re-anneal back. Thus, they have to be kept apart by a set of proteins called SSB, single-strand DNA binding proteins. This is what we refer to now as the replication bubble, while each side is called the replication fork, the reference point that we shall be using in our discussion later on. In our books and references, what is usually illustrated to depict replication is just one side of the replication bubble. That's why it looks like the letter Y in our books and references. So for now, we'll discuss the event using that point of view. So let's enlarge that side of the replication bubble. Let's imagine the helicase continuing to separate the two parental strands from each other. SSB binds to the single strands to prevent them from re-annealing. So now we have here the parental DNA strands ready to serve as templates, but first, primers have to be laid down by the enzyme primase. Now remember, primers are not composed of DNA, but are rather composed of RNA. So the enzyme primase can also be referred to as an RNA polymerase because that's what they do. They polymerize RNA. Now, once the primers have been laid down, the 3' end of the primers contain a hydroxyl group, which can be targeted by DNA polymerase and incorporate nucleotides to polymerize DNA in the 5' to 3' direction, as represented by the purple arrows. So far, do you have any question? Do you all follow?
No questions po.
Okay, so let's imagine the helicase continuing to separate the two strands once more. Let's focus our attention on the top strand. The 3' end here contains hydroxyl group. The DNA polymerase will simply attach nucleotides here, resulting to the polymerization of DNA. Now let's go to the bottom strand. This is where it becomes a little more complicated. Question: can we incorporate a nucleotide at the 5' end here? Yes or no?
No po.
No, tama. Kasi wala namang hydroxyl group yan. So the problem is, how do we now synthesize DNA here? That's a problem. So the solution to overcome the problem is to lay down a new set of primers. Once primers have been laid down, the 3' hydroxyl group here will serve as a target for DNA polymerase so that it can incorporate nucleotides to polymerize DNA, as always, in the 5' to 3' direction. Now let's Imagine helicase continuing to separate the two parental strands once more here at the replication fork. For the top strand right here, the DNA polymer simply targets the 3' end to incorporate nucleotides so that there will be polymerization of DNA. While here, as always, a new set of primer is laid down, after which it will be extended by DNA polymerase. Let's go to the top strand. This is referred to as the synthesis of the leading strand, while here at the bottom, this is referred to as the synthesis of the lagging strand. Let's characterize the synthesis of the leading strand. You can turn on the microphone and answer. First question, leading strand, does it require primers? Yes or no?
No po, Doc.
Ano yung kulay green doon? So, ano? Revise your answer, yes or no?
Yes po. Required.
Yeah. Yes, laging required. Hindi pwedeng gumawa ng DNA kung walang primer. What is the direction of polymerization for the leading strand?
5' to 3'.
That's right. Is the process continuous or discontinuous?
Continuous.
Okay, and what is the direction of growth? Towards the replication fork or away from the replication fork?
Forwards.
Okay, and what is the main enzyme for the leading strand synthesis? There are two DNA polymerases. Is it DNA polymerase 3 or is it DNA polymerase 1?
3.
3. Very good. Now let's move on to the lagging strand synthesis. Let's also characterize it. First question, does it require primers?
Yes.
Yes, just like the leading strand, pero mas marami siyang primers. Next question, what is the direction of polymerization for the lagging strand?
5'.
5' to 3'.
That's right, just like the leading strand. Is the process continuous or discontinuous?
Discontinuous.
That's right. Because it is discontinuous, it results to the formation of individual Okazaki fragments. So this is an Okazaki fragment, and so is this. So you can say that Okazaki fragments make up the lagging strand, or the lagging strand is composed of several individual Okazaki fragments. Now, what is the direction of growth of the individual Okazaki fragment? Is it towards the replication fork or away from the replication fork?
Away po.
Away.
Right. Okay, final question. If the leading strand's main replicative enzyme is DNA polymerase 3, what is the main replicative enzyme for the lagging strand?
3 din po. Same
That's
lang.
right. Yeah, so DNA polymerase 3 is the main replicative enzyme for both the leading and lagging strand. The question is, what is the role then of DNA polymerase 1? Why did we mention it? Let's focus here on this Okazaki fragment primer. Is the primer RNA or DNA?
RNA po.
RNA. So eventually it has to be taken out and replaced with DNA. And that is the job of DNA polymerase 1. So let's focus our attention here at the 5' end of that primer. It was digested, and the enzyme that digested that is DNA polymerase 1 through a 5' to 3' exonuclease enzymatic activity. This leaves us with a gap that is devoid of DNA and devoid of primer. So hindi yan pababayaan ng DNA polymerase 1. DNA polymerase 1 will target the 3' hydroxyl group here, and through its 5' to 3' polymerase enzymatic activity, it will incorporate nucleotides to the 3' end so that it can synthesize DNA. So habang tinatanggal niya yung primer, nagsisynthesize siya ng DNA. After the primers have been taken out, the individual fragments are still discontinuous. So there's a third enzyme we did not mention earlier. This is called DNA ligase. Using energy from ATP, it connects adjacent Okazaki fragments together so that it's one single continuous strand without any breaks. So, naintindihan? Okay, that's good. Let's repeat and review to see if you really understood what is going on. So we have a prokaryotic closed circular DNA. The process of replication takes place here in the origin of replication. So we're going to zoom in. The first event is that the two strands are separated from each other by the enzyme helicase, and the two strands are kept apart by these proteins. What do we call these proteins?
Single strand binding protein.
Okay, this is called the replication bubble, and each side is called the replication fork. Earlier, we saw what was going on the side of the replication bubble. This time, let's take a look at both. Helicase separates the two replication forks. Primers are laid down by the enzyme RNA polymerase or primase. After which DNA polymerase 3 targets the 3' ends of the primers, incorporating nucleotides to be able to polymerize DNA in the 5' to 3' direction. This right here is what we saw earlier. Now, we're taking a look at both sides. Okay, so question, there are 4 fragments here. Which one are the leading strands? Let's say this is number 1, number 2, number 3, and number 4. Sino dyan ang leading strand? Anyone? Ulitin, ano dyan ang leading strand? 1, 2, 3, 4. Which one is the leading strand?
Parang wala po yata.
Parang wala. Meron.
2 and 4 po, Doc.
That's right. Very good. So number 2 and number 4 are the leading strand because they're growing towards the replication fork. This is the leading strand we saw earlier. Its counterpart on the other side is this. It's also growing towards the replication fork. So this would be the leading strands. You can see they're growing towards the replication fork. While this would be the lagging strand that we saw earlier, its counterpart on the other side would be this. Did we violate the 5' to 3' rule here? No, it's still consistent. 5', 3', 5', 3', 5', 3'. So it's consistent. The same would be true for the ones located at the bottom. Now, let's focus our attention on the 2, replication fork. As you can see, they are moving away from each other. What do we call that direction if those 2 replication forks are moving away from each other? Do we refer to it as unidirectional or bidirectional?
Bidirectional.
That's right. So the 2 replication forks are bidirectional. Now, eventually, those Okazaki fragments will be taken out and replaced with DNA by the enzyme DNA polymerase 1. And then the fragments are going to be connected together by the enzyme ligase. So let's take a look at replication overall. So we started here. This is the origin of replication in a closed circular DNA. This is the replication bubble itself that formed in the origin of replication. And each side is called the replication fork. The 2 replication forks are moving away from each other, so they are said to be bidirectional. Do you think they will meet at the other end? Those 2 replication forks, yes or no? Makikita rin ba sila sa huli at sa dulo?
Yes po.
Yes, okay. Parang pag-ibig, magkahiwalay man kayo, sa dulo kayo pa rin magkikita. So those two replication forks are going to meet at each other because it's bidirectional and it's a closed circular DNA. When they do meet at each other at the other end, that's the end for replication. Termination yon. The result is that we now have two identical closed circular DNA. Replication successful. Now let's discuss the enzymes involved in replication in prokaryotes. So there are three DNA polymerases, 1, 2, and 3. We have discussed DNA polymerase 1 extensively, and it is involved in taking out primers and replacing primers with DNA. So we won't discuss it anymore. DNA polymerase 2 is involved in mitochondrial not mitochondrial DNA repair. So hindi na natin yan discuss. While DNA polymerase 3 is the main replicative enzyme. So let's discuss this one extensively. DNA polymerase 3 is the main replicative enzyme. And just like DNA polymerase 1, it has a 5' to 3' polymerase enzymatic activity. This is the enzymatic activity that allows those two enzymes to synthesize phosphodiester bond. To illustrate, let's say this is a template. The goal is to synthesize a new strand. But before DNA polymerase 3 can exert its 5' to 3' polymerase function, there should be a primer already waiting. And then DNA polymerase 3 will target the 3' hydroxyl group here and exert its 5' to 3' polymerase activity. Okay, so it's pretty much straightforward. Now, aside from the 5' to 3' polymerase activity, DNA polymerase 3 has another enzymatic activity. So to illustrate what that is, let's start from a clean slate. So we have a template. Our goal is to synthesize a new strand. Primers are laid down first, and then DNA polymerase 3 will come in and exert its 5' to 3' polymerase enzymatic activity. Ngunit, subalit datapwan, Diyosa man siya, nagkakamali rin. That's Dr. Vibar's favorite expression. Diyosa man daw siya, nagkakamali din. That would be true for DNA polymerase as well. You see, it has incorporated the wrong base, resulting to a mismatch. So titigil muna siya sa kanyang 5' to 3' polymerase activity and will shift to a different enzymatic activity. This is known as the 3' to 5' exonuclease. Bakit 3' to 5'? Aatras siya, babalik. And why exonuclease? It will digest the phosphodiester bond of the mismatch. So that mismatch is removed. Now, after that mismatch has been removed by this enzymatic activity, DNA polymerase 3 will now revert back to its original 5' to 3' polymerase activity. And hopefully this time, hindi na siya magkakamali. So the 3' to 5' exonuclease activity of the enzyme is referred to as the proofreading function of the enzyme. Bakit proofreading? Pang-erase ng mistake. What do you think would be the consequence if DNA polymerase does not have a proofreading function? Anyone?
Mutation.
That's right, mutation. Mistakes are often made and they are not corrected. So the proofreading function minimizes, not totally eliminates, minimizes the occurrences of mismatches. As a matter of fact, RNA viruses have RNA as their genetic material. RNA viruses uses RNA polymerase to make a copy of their genetic material. RNA polymerases do not have a proofreading function. That is why RNA viruses often have high rates of mutation every time they copy their genetic material. Anong RNA virus ang sikat ngayon? Anyone? It's always in the news. It's the corona virus. Do you ever wonder why it's constantly mutating? Because it uses RNA as its genetic material and RNA polymerase to make a copy of that RNA. It does not have any proofreading function. Kaya, every time it copies, lagi at laging magkakaroon ng mutation. It's inevitable. It's unavoidable. Let's summarize now the two enzymes. DNA polymerase 3 is the main replicative enzyme as it polymerizes both the leading and lagging strand through a 5' to 3' polymerase enzymatic activity. And it has a proofreading function to ensure fidelity of replication. On the other hand, DNA polymerase 1 takes out the primers. How? By a 5' to 3' exonuclease activity. This is different from this. Then it replaces the primers with DNA via the same polymerase activity as this one. And just like DNA polymerase 3, it also has a proofreading function to ensure fidelity of replication. Let's move on now to eukaryotic DNA replication. Has a lot of similarities with prokaryotic DNA replication with some major differences. So what are the similarities? Eukaryotic DNA is double-stranded, so both parental DNA serves as template, just like in prokaryote. It's also a semi-conservative process. However, unlike prokaryote, there are multiple origins of replication and not just one. Eukaryotes have a cell cycle, so the process of replication occurs only during the S phase. Now, for humans, the number of DNA that we have is 2n. So during G1, the number of DNA or chromosome is 2n, and at S phase, where replication happens, it doubles up to 4n. And this will be restored back to 2n after mitosis. So just a visual comparison between prokaryotic versus eukaryotic DNA replication. In prokaryote, the DNA is double-stranded, close circular, and small, and there is just one single origin of replication. In eukaryote, each chromosome is composed of large, long, double-stranded linear DNA. And there are, of course, multiple origins of replication that occur during the replication process. What about the enzymes? In prokaryote, there are 3 DNA polymerases, with DNA polymerase 3 being the main replicative enzyme for both the leading and the lagging strand. In eukaryote, 5 DNA polymerases have been identified. These are the ones that you need to remember. DNA polymerase alpha is complexed with the enzyme primase, and they are the ones that create the primers. DNA polymerase epsilon is the dedicated enzyme for leading strand synthesis, while DNA polymerase delta is the dedicated enzyme for the lagging strand synthesis. So there's a separate enzyme for the leading and the lagging strand synthesis in eukaryote. But the principle is the same. Helicase separates the 2 parental strands. SSP binds to the single strands. Primers are laid down by the enzyme primase, but then primase is complex with DNA polymerase alpha that incorporates a short segment of DNA, after which the DNA polymerase alpha primase falls off, giving way to DNA polymerase epsilon for the leading strand and DNA polymerase delta for the lagging strand. Let's summarize now replication by comparing prokaryote versus eukaryote. So both are semi-conservative. Both are primer dependent, which means it cannot be initiated without primers. But the primers in prokaryote are synthesized by the enzyme primase, while in eukaryote, primers are created by the complex of primase and DNA polymerase alpha. So both strands, parental strands, serve as template in both prokaryote and eukaryote. But there's a single origin of replication in prokaryote and multiple origins of replication in eukaryote. DNA polymerase 3 creates the leading and lagging strand in prokaryote, while in eukaryote, delta and epsilon for the lagging and leading strands, respectively. What about the end replication problems? In prokaryote, there's the formation of supercoil, which is also observed in eukaryote. There's one problem unique in eukaryote, and that is telomere shortening. So finally, what are these end replication problems? Why do they occur, and how can they be solved? The first end replication problem is associated in both prokaryotic and eukaryotic replication, and that is the formation of supercoil. To illustrate the concept of supercoil, let's imagine a landline cable. So the cable is coiled. When it coils upon itself over the coil, that is now the supercoil. So imagine if this is DNA, the coil would be the helix, and then it coils upon itself, giving supercoil. Now, closed circular DNA is said to be relaxed when you can compare to a bracelet, which can be laid down flat on a table. And this is the supercoiled version. So it twists upon itself on top of the superhelix. So why does supercoiling occur during replication? It requires a bit of imagination. So imagine if we're going to separate the two strands here. You're going to separate them. Since this is closed circular, the helices here would tighten if we loosen the helix here. Because, well, it will twist and it has nowhere to go. So if this tightens, this entire DNA will coil upon itself, producing now the supercoil. Why is that a bad thing? The supercoiling produces a lot of strain in the sugar phosphate backbone and can result to the random breakage and damage in the DNA. So it has to be addressed. So supercoiling can be solved by the enzyme called topoisomerase. How does it work? It cuts the sugar phosphate backbone to relieve the stress produced by the supercoiling and then reconnect back the sugar phosphate backbone. There are two types of topoisomerase. Type 1 creates a single cut on a strand, then reconnect. Type 2 creates a double cut on the two strands. One cut on one strand and the other on the other strand. Release the tension and then reconnect. To illustrate, we have here type 1 topoisomerase. So if you take a look at this closed circular DNA, it has 3 negative supercoils. Once the topoisomerase has acted on it, it is now partially relaxed. There are only now 2 negative supercoil. So how was type 1 topoisomerase able to accomplish that? Let's say we are zooming in into this segment that has been enlarged here. So there are 2 strands here that are intertwined, the red and the blue. And in this particular section, the red is on top of the blue. The topoisomerase will cut the red strand. Then it will reconnect the red strand, this time behind the blue strand. As a result, the helix is now looser. This results to the overall relaxation of the entire closed circular molecule. So it is now partially relaxed with less negative supercoil. The last end replication problem is telomere shortening that you have discussed earlier this morning in your CBL. And this is a problem exclusively found in eukaryotes. Why does it shorten? Telomere refers to the ends of the linear DNA. So let's imagine that this is the parental strand separated from each other during replication, while this is the primer and the DNA strand, the leading strand. Now here we have the lagging strand. This is the primer, and this is the DNA. This would be the last Okazaki fragment of the lagging strand. As in our earlier example, this primer can be taken out and replaced with DNA because DNA polymerase can simply target the 3' hydroxyl group here and incorporate nucleotides to synthesize DNA. Now let's go to the last primer. It will be taken out. But it cannot be replaced with any DNA because, well, there's nothing here. Meron ba tayong 3' hydroxyl group dito na pwedeng targetin para maka-incorporate ng nucleotide? Wala. As a consequence, it is now shorter, and these DNA sequences have been lost. We have here a 3' overhang. So imagine if this is going to undergo another round of replication, that telomere will become shorter and shorter and shorter. So for eukaryotes, there's only a certain number of times that DNA can be replicated, after which they have to stop. Otherwise, nothing would be left. But for some cells, they do have an enzyme that can address telomere shortening. That enzyme is called telomerase. What is unique about that enzyme? It has, well, aside from the protein part, it has a built-in RNA template. And what will the RNA template do? It will bind to the 3' overhang, and the enzyme right here will incorporate a nucleotide to the 3' end of this overhang using the RNA as the template. What do we call that process where DNA is synthesized using RNA as a template? Anyone?
First transcription po.
Yes. So telomerase is actually an ancient form of reverse transcriptase. The story does not end here. When the telomerase goes away, a new set of primer can be laid down, after which it can be targeted by DNA polymerase so that whatever sequences that have been lost have now been fully restored. But what if we take out the primer here? Meron naman tayong sobra. Well, I won't be answering that because that has already been answered in your CBL this morning, but telomere regulatory proteins would bind to it and seal this so that it will not be targeted by exonucleases. So question, what are these cells? Not all cells have active telomerases, right? This has been discussed early this morning. Somatic cells do not have an active telomerase. What are the cells that do have active telomerases? Anyone?
Progenitors.
Yes, cancer cells. Ano pa? Progenitor, stem cells, germ cells. These cells are capable of infinite division without the worry of shortening of telomere because they do have telomerase. But the regular somatic cells do not have an active telomerase. So for humans, and just like all other telomerases, the telomerase contains two subunits. The enzyme part is reverse transcriptase enzyme, and it has a built-in RNA template. So let's summarize before we go on a short break. DNA is the repository for genetic information, as it is the genetic material. It can be replicated in a semi-conservative manner. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously. Now, telomere shortening and supercoiling are end replication problems telomere shortening can be addressed by telomerase, this is a problem exclusively seen in eukaryotes, while supercoiling is a problem seen in both, but it can be solved by the enzyme topoisomerase. So let's have a short break, 5 minutes, after which we shall continue with the second part of our lecture. Right, so can you see the presentation?
Yes po.
So for the second half, we shall be discussing now the last two processes, both transcription and translation. So we'll discuss first, as always, prokaryotic transcription before we go to eukaryotic transcription. And of course, for eukaryotic transcription, we shall be focusing on the modification that takes place. And the last event that we shall be discussing is protein translation. So let's start now with prokaryotic transcription. Transcription in general is defined as the synthesis of RNA with DNA as the template. Now take note that certain stretches of DNA are, only certain stretches of DNA can be transcribed. These are the ones that are referred to as genes. So in other words, all genes are DNA, but not all DNA can be called genes. Only those that can be potentially transcribed are referred to as genes. The enzyme for transcription is RNA polymerase, and the product would be all forms of RNA, such as mRNA, rRNA, and tRNA. For today's discussion, however, we shall be focusing on the transcription of mRNA as the representative example. So let's have an overview first. So let's say this is a DNA. Just like replication, the first event is separation of the two DNA strands. But unlike replication, only one of the two strands can be used as template for the synthesis of RNA. And RNA is polymerized in the 5' to 3' direction in a manner similar to how DNA is polymerized. So this DNA strand is referred to as the template, the non-coding or the antisense strand, while this DNA strand right here is referred to as the non-template coding or the sense strand. So we can divide transcription generally into 3 stages, just like how we divided transcription, replication. So that includes initiation, elongation, and termination. Now in prokaryote, whatever RNA is being transcribed, there's only one single type of RNA polymerase for the transcription of all these different types of RNA products. And in prokaryote, transcription and translation are coupled together. What does that mean? So let's say we have here a prokaryotic organism. So the DNA is being transcribed by an RNA polymerase, and the mRNA, even before it is fully transcribed, can already be used by the ribosome for translation. So the RNA does not have to be completed for translation to begin. So what is the reason for that? Well, the RNA does not have to be modified. It's ready to be used. And number 2, there is no physical separation between the DNA and the rest of the cytoplasm in a prokaryotic organism. The enzyme for transcription is the RNA polymerase. As the model shows here, it is composed of the core enzyme, plus there is the detachable sigma factor. So they have to be together initially for them to locate and bind the gene that needs to be transcribed. The 5' to 3' polymerase function resides in the core enzyme, while the sigma factor is regulatory in function. Now, just like what I have said earlier, RNA polymerases are devoid of 3' to 5' exonuclease function. That means they are prone to making mistakes. And pharmacological correlation, drug for tuberculosis like rifampicin exerts its antibacterial effect by inhibiting RNA polymerase on mycobacterium. The segment of DNA that is going to be transcribed is called the gene, and it has the following parts. There is the promoter region, which is the initial binding site for RNA polymerase. Now, there are different types of promoter regions. Some are called the Pribnow box, the GC box, Tata box, Hugnes box, and so on. These are all located close to the informative sequence, which is the actual transcriptional site. Now, there are two strands. Only one strand will serve as template. As you can see here, only one of the two strands will be used as template for the synthesis of RNA. The last part of the gene is called the terminator sequence. So this dictates the endpoint for transcription. As you can see here, it's a GC-rich region followed by a poly-A tract. The presence of a poly-A tract actually determines how transcription ends. If the poly-A tract is absent, termination will require a terminator protein called a Rho factor. So let's go to the transcription process itself, starting with initiation. The RNA polymerase holoenzyme would locate and bind the promoter region, forming now what is known as the closed promoter complex, after which the sigma factor can now dissociate. The RNA polymerase does everything, unlike the DNA polymerase, which requires several other enzymes. So the RNA polymerase even acts as a helicase, separating partially the two DNA strands from each other, forming now what we call the open promoter complex. Then it moves forward, locating the informative sequence. Once the informative sequence has been located, it will now start polymerizing RNA de novo. Ibig sabihin, wala ng primer. Kasi yung primer RNA din naman eh. So, diretso na siya. So, it polymerizes RNA in a 5' to 3' direction, just like how DNA is polymerized. And it will continue polymerizing until it reaches the terminator sequence. Now, even before the RNA polymerase has reached the terminator sequence, ribosomes can already bind to the 5' end of the RNA and use it as a template for translation. That's why we say transcription and translation are coupled together in the prokaryote. As a matter of fact, several ribosomes can bind to the RNA. You can imagine a polypeptide coming out of the ribosome, but I did not put that anymore for the sake of simplicity. In other words, tinatamad na mo ng animation. So let's imagine that the RNA polymerase has reached the terminator sequence. This will now dictate the endpoint for transcription. So the sequence is GC, so we would expect the complementary sequence to be found on the RNA, CG. The C on the RNA would be attracted to its own G. So kumbaga, gandang-ganda siya sa sarili niya. It will hybridize with itself, forming a hairpin loop because of self-complementarity. What follows next is A. What do you think would be the sequence on the RNA if you have A on the template? Anyone?
Polyuracil.
That's right. U, and U. How many hydrogen bonds are there between U and A?
2 hydrogen bonds.
Okay, that means their bond is very weak. Because their bond is very weak, the RNA is easily displaced from the template, ending now transcription. So parang relasyon, no? If your bond as a couple is not that strong, the usual cause of breakup is, well, you just naturally drift apart for no apparent reason. Nagkahiwalay lang, basta-basta. But if your bond as a couple is very strong, the usual cause of breakup is a third party. That would also apply to transcription. So as always, there's the formation of hairpin loop, but there's no poly A track. So there could be a G, there could be a C, and so on. So the bond between the RNA and the DNA template is still very strong. This is where the third party will come in the form of a rho factor. It will act as a helicase, breaking the hydrogen bond between the RNA and the DNA template, causing the RNA to dissociate from the template, ending now transcription. Sabi nga, talo ang magandang Diyosa sa malanding impactita. That would also apply to your relationship. In English, the evil temptress beats the gorgeous goddess. So natatawa kayo, which is great news for the guys. This could be your motivation in becoming a doctor because, no matter how ugly you are, I'm not saying that you are, but no matter how ugly you are, girls would be fighting over you once you become a doctor. So that could be your source of motivation. So let's summarize now prokaryotic transcription. RNA polymerase catalyzes the formation of RNA. The process begins by locating the promoter sequence and it ends when the terminator sequence is reached. And there are two ways to end. More commonly, it's rho independent and less common is the rho dependent termination. mRNA is ready to be used even before it is fully transcribed. Let's move on now to eukaryotic transcription. Of course, it is expected to be much more complex, and there are three classes of RNA polymerase for the different types of RNA. Initiations require several other proteins, not just the sigma factor, and it can be influenced by several other sequences, not just the promoter region. The RNA undergoes extensive modification. So the example that we shall be using here is the mRNA as well. Now, in eukaryote, transcription and translation are not coupled. What's the reason for that? Transcription, which occurs in the nucleus, produces an RNA that has to be modified first before it is transported into the cytoplasm for translation. And number two, there is, of course, a physical separation between the nucleus where transcription takes place and the cytoplasm where translation occurs. What about the eukaryotic gene? It has the same basic parts, but it is, of course, more complicated. The basic parts includes the promoter region, which is, as always, the attachment site for RNA polymerase. Now, this promoter region can be influenced by other DNA sequences. These DNA sequences can be called enhancers. So these are just like promoter regions, but are located far away from the informative sequence. The informative sequence, as always, is the actual transcriptional site. But within the informative sequence, there are coding and non-coding regions. The coding regions are referred to as exons. They are sequences that are transcribed and ultimately translated into proteins. These exons are interrupted by intervening sequences known as introns, non-coding regions within the informative sequence. These are transcribed but are not translated because they are taken out in the resulting RNA. And the terminator, as always, ends transcription. Okay, now the RNA polymerase in eukaryote are of three types. There's RNA polymerase 1, which transcribes ribosomal RNA with the exception of 5S. 2 transcribes mRNA, and 3 transcribes tRNA, including 5S. Easy to remember. 1, 2, 3 corresponds to R, M, T. As you can see from this illustration as well, the RNA polymerase does not work alone. It requires several other proteins to form the initial eukaryotic transcription complex. What are these other proteins? This would include activators, shown in red. They bind to the enhancer and bring the enhancer close to the transcription site and also help position the RNA polymerase properly. Now, activators can be antagonized by repressors, shown here in blue. Now, as a matter of fact, the interaction between activators and repressors help regulate transcription. The basal factor, shown in green, is the equivalent of the sigma factor. They bind to the promoter region and they position the RNA polymerase properly. Finally, there's the transcription factor, shown in gray. So they also position the RNA polymerase properly. Once positioned, the RNA polymerase is phosphorylated as what occurs in the prokaryote, and then is released and does its job in a manner similar to what we have already seen in prokaryote. It acts as a helicase, separating the two strands. One strand is used as a template for the polymerization of mRNA. The process continues until the terminator sequence is reached. Now, what is unique about eukaryotic transcription is that the RNA undergoes extensive modification. This would include addition of a 7-methyl guanosine cap, splicing, and addition of a poly-A tail. To illustrate this modification, so let's say this is the informative sequence of the gene and it has been transcribed into an mRNA primary transcript. The first modification is addition of a 7-methyl guanosine cap at the 5' end. The cap actually allows for proper ribosome binding later on during translation. The next modification is that the introns that are found on the mRNA are spliced out, leaving only behind exons. This results to an mRNA that is considerably shorter compared to its original template. And the final modification is addition of a poly-A tail. This is to allow proper ribosome binding as well during translation. This is now the mature mRNA that will be translocated into the cytoplasm for translation. How do we regulate transcription? There are different mechanisms for regulating transcription. Some of them are beyond the scope of what we can discuss today, but you can read on them. This would include acetylation of nucleosome, methylation of CpG islands, the interaction between activators and repressor binding to promoters, operators, and enhancers, and alternative splicing. We shall be discussing this too. Let's start with alternative splicing. You might be wondering, why do we have exons and introns if we're going to splice out introns anyway? Well, the presence of introns allow what is known as alternative splicing that provides a lot of versatility to the limited number of genes that we have. For example, in one instance, exon 1, 2, and 4 can be spliced together to come up with one mRNA variant that can give rise to one type of protein. In another instance, exon 1, 3, and 4 can be spliced together to come up with another mRNA variant that will give rise to a totally different type of proteins. So for example, humans have about 30,000 genes, very limited number of genes, and yet humans are capable of producing more than 100,000 different types of proteins. How can we produce that many number of proteins if we only have a limited number of genes? Well, the answer to that is alternative splicing. Another means of regulating transcription is illustrated by the lac operon. This is not new to you because it has been introduced in your first lecture last sem. So the lac operon is a series of genes. One of them codes for the enzyme lactase, and it is under the control of other sequences such as the operator, which can be likened to the off switch, and the promoter region, which of course is the on switch. So let's say that the organism does not have access to lactose, which means the lac operon has to be shut down. Without lactose, a protein repressor would be present. It binds to the operator, shutting down the lac operon because the RNA polymerase is unable to bind to the promoter region. If the RNA polymerase does not bind to the promoter region, there would be no transcription of this. In effect, it is rendered silent. But what if the sugar lactose is now present? Lactose binds to the repressor. This causes a conformational change on the repressor so that it loses affinity to the operator. This will now would allow the RNA polymerase to have free access to the promoter region. The RNA polymerase, of course, would now transcribe the lactate gene into mRNA, and the mRNA is translated into the protein lactase. So finally, let's summarize transcription. So it is in eukaryote, of course, it's expected to be much more complex. The process begins by locating the promoter region, but it can be influenced by other DNA sequences such as enhancers. And there are three classes of RNA polymerase, 1, 2, 3, that transcribes RMT. RNA is extensively modified. And what are these modifications? Capping, splicing, and tailing. And finally, we have reached the final process. Hooray. This is translation. It is defined as the synthesis of protein polypeptide using mRNA as the template, and the process occurs in the ribosome. The minimum requirements include the tRNA, the rRNA, and the mRNA. The process starts with the start codon, AUG, which also codes for methionine, and ends with the stop codons. And there are 3 stop codons that can be used. So let's have an overview first. We all know that these are the products of transcription. They will all come together at the ribosome, where translation takes place to produce a protein. What are the roles of these different RNA molecules? mRNA, of course, determines the amino acid sequence by acting as a template. The tRNA is a carrier for that amino acid that will be incorporated into the polypeptide, while rRNA is provides the enzyme that catalyzes peptide bond formation and also forms the ribosome. So this is a simple version of translation. It starts with AUG that is located near this start codon. It also codes for methionine. The next codon specifies now the sequence of amino acids that will be incorporated into the polypeptide, with the peptide bond synthesized by the enzyme peptidyl transferase. The process ends with a stop codon. Any of the 3 stop codons may be used. If you notice, the polymerization of the polypeptide occurs from the N to the C terminus. We can divide translation into 4 major steps. This includes pre-initiation, where the tRNA are charged, or in other words, the amino acids are going to be attached to its carrier. That is the tRNA. Initiation is where the start codon is going to be located, followed by elongation. So the amino acids carried by tRNA enter the ribosome via the A site. This is followed by peptide bond formation and a movement called translocation. And finally, termination is when the stop codon is finally reached. So let's start with pre-initiation. All amino acids that will participate in translation have to be energized via adenylation with ATP. Once adenylated, these amino acids will be attached to their carrier, and that is the tRNA, by attaching them to the acceptor arm of the tRNA. And the process, of course, is catalyzed by an enzyme called aminoacyl tRNA synthetase. Now let's move on to the initiation. We need the ribosome. The small ribosomal subunit has to be separated from the large ribosomal subunit. It has the P site, which is the entry point for the initiating amino acid, whereas all other amino acids have to enter via the A site. The tRNA that carries methionine will enter the P site. This will scan the mRNA so that the P site is aligned with the start codon, and the next codon is aligned with the A site. Last to join is the large ribosomal subunit. So it forms the complete 70S initiation subunit. It will be phosphorylated and ready to move on to the next phase, which is elongation. So UCU, aligned with the A site, is ready to accept the next codon, which is serine, carried by tRNA. The thionine from the P site will be released and will form a peptide bond with serine. The process of peptide bond formation is catalyzed by the enzyme called peptidyl transferase, a ribozyme that is found here in the large ribosomal subunit. So this tRNA is now empty and will move away from the P site. The P site is now vacant. The next codon is CGU, which codes for arginine. But before it can accept arginine, it has to be aligned with the A site. So there is a movement called translocation. So the A site is now aligned with the next codon, and the dipeptide is now carried by the P site. So the tRNA that carries arginine enters the A site. So as always, there would be peptide bond formation catalyzed by peptidyl transferase. And this empty tRNA will move away from the P site, and this would be followed by translocation so that the polypeptide is held by the P site and the A site is aligned with the next codon. So really, translation is just a repeated cycle of peptide bond formation followed by translocation, peptide bond formation followed by translocation, repeated over and over again till the A site is aligned with the stop codon. When the A site is aligned with the stop codon, there is no tRNA for this or an amino acid, but rather a release factor binds to the A site. This causes the polypeptide to dissociate from the complex, ending translation. What happens now to the polypeptide? Well, it can undergo post-translational modification that are already beyond the scope of what we can discuss today, but you can read about it. What happens to this ribosome? It will dissociate from each other and can be used again, while the mRNA can be used several times for translation. Now, after some wear and tear, this RNA eventually will be destroyed. So let's summarize translation and end today's lecture. It is defined as the synthesis of protein or polypeptide using mRNA as the template, and the process begins with the start codon, AUG, which also codes for methionine, or formulated methionine in case of prokaryote. Translation is described as a repeated cycle of peptide bond formation followed by translocation, with the peptide bond synthesized by the enzyme called peptidyl transferase, a ribozyme found on the large ribosomal subunit. And the process of translation ends in the stop codon. Any of the 3 stop codons may be used. So that ends our lecture for today. Finally, this is the last one. All right, so do you have any questions? Wala na, tulog na sila. If there are no questions,